Okay, welcome back. So now we're moving on to the topic of proteins. So we are moving on to the topic of the proteins. Where is the chat? Chat. Oh, okay, audio. Are we live? This one is live. Okay. Good evening, friends. Okay, just confirm that you are able to see the video and you are able to hear the audio, and then we'll start. Just confirm that you are able to see the video, the audio, and the screen. Ah, great, great. It is good to hear that you are able to clearly see the video, audio, and the. Okay, so we are moving on to proteins. We are moving on to proteins. Before I go on to proteins, I would like to ask one thing. How do you define? When you try defining, you will have to distinguish between peptides, polypeptides, and proteins. So how do you distinguish between the peptides, polypeptides, and the proteins? Okay, so how to distinguish between the three peptides will be less than equal to 50 amino acids, whereas greater than 50 amino acids will be polypeptides, and proteins will be greater than 100 amino acids. So basically it is based on the number of amino acids. Please note that this distinction is quite arbitrary. There is no structural reason why we should call them peptides, polypeptides, or proteins based on the number of amino acids. So this is arbitrary, and we can slightly deviate from these numbers. For example, the insulin, 51 amino acid peptide. It should be called polypeptide, but we still call it the peptide hormone. We call it the peptide hormone. So the numbers are just to give us a rough idea of the likely structure that we are dealing with. Now, in the past, various classifications have been used to discuss the proteins. For example, the integral and the soluble protein, the globular and the fibrillar protein, but they don't hold much significance in terms to the function of the protein. So, the function of the protein, the function of the protein depends on the organization of the protein. So nowadays what we focus on is the four orders of protein structure. What we focus on is the four orders of the protein structure. This will help us to determine. Oh yes, you're right. You're right. Okay, no problem, no problem. We'll sort it out. Don't worry, don't worry. I'll on to it. Let me see. We'll reinstall it or restart it. Okay. I hope now you can see this. Is this visible to everyone now? Is it visible to everyone now? I presume it should be visible now. Okay, we'll start from the beginning, no problem. So I said that now we're going to be talking about proteins, and before you go into the details, you should know what do you mean by proteins. So obviously, proteins are amino acid linked together. So when we talk about amino acids being linked together, we have three different structures which come to mind: the peptides, the polypeptides, and the proteins. So all of them are representing the amino acid linked together. So how do you distinguish between them? Remember, the peptide have up to 50 amino acid. Polypeptide will have more than 50 amino acid. A polypeptide cannot be called a protein until unless it has more than 100 amino acid. A polypeptide cannot be called a protein until unless it has more than 100 amino acids. Alright, so the peptide, polypeptide, and protein tell us the approximate size of the molecule that we are dealing with. Please note that these are very arbitrary classifications, and I've given the example of insulin. The insulin having 51 amino acid, and it is still called a peptide hormone. It is still called a peptide hormone. So these names are just to give us a rough idea of the structure of the molecule that we are dealing with. They do not have any implication on the function of the protein. They don't have any implication on the function of the protein. Now, in the past, various different classifications have been used to discuss the protein. For example, one classification says soluble and integral protein. This basically deals with the proteins which are present in the membrane, soluble and integral protein. Another classification says globular and a fibrillar protein, depending on the shape, whether it is looking like a spherical structure or it is a elongated protein. So globular and fibrillar protein. However, these do not exactly describe the function of the proteins. Finally, it has been understood that the function of the protein depends on the shape of the protein. The function of the protein depends on the shape of the protein, and therefore we focus on the organization, how the protein is organized and what is the final shape that we are able to see. So the current discussion of the protein will focus on the organization of the protein at different levels, and these are the primary, secondary, tertiary, and quaternary levels of the protein organization. The different levels of protein structure. The first, second, third, fourth, also known as primary, secondary, tertiary, and quaternary structure. Okay, so moving to the first, the primary structure. As you can see from the definition, it is the sequence of amino acid in a polypeptide chain. It's the sequence of amino acid in a polypeptide chain, just which is the first, second, third, fourth amino acid, whichever is there. So the sequence of amino acid in the polypeptide chain. Now, some amino acids which are close together, they tend to start folding as per their structure, and they form localized specialized shapes. These localized shapes of contiguous, the amino acid which are coming one after the other, is the contiguous amino acids is known as the secondary structure. So we have the primary structure, then we have the secondary structure. Here, the contiguous segment of polypeptide will fold into well defined shapes. They will fold into well defined shape. Remember, these are the contiguous amino acid. Some examples of the secondary structure can be alpha helix or the beta pleated sheet. Alpha helix or the beta pleated sheet. Alpha helix or the beta pleated sheet. All right. When we have a large number of secondary structures, they can come together in three dimension and form the final shape of the protein. So final shape of the protein depends on the clustering and the arrangement of all the secondary structures. This is what we call the tertiary structure. The tertiary structure is assembly of all the secondary structural units in the mature polypeptide. In the mature polypeptide, this is the tertiary structure. In tertiary structure, we may find distant amino acids are coming close. Distant amino acids may come close in the tertiary structure. Distant amino acids may come close in the tertiary structure. So you can compare it like when you join the college in the on the first day, you only have the roll number of the students. On the first day, you only have the roll number of the students. But after a few days, the students who have the similar roll number, the students who have the similar roll number, they will start forming some good rapport among themselves. So that is something like the secondary structure. And after some time, the whole class will have some defined interaction between them, which is the tertiary structure. Now, in some cases, the protein can have more than one polypeptide. For example, hemoglobin, four polypeptide are there. We had seen yesterday LDH. LDH has also four polypeptide. In the creatine phosphokinase, we had seen the two polypeptide are there. So, when more than one polypeptide are there, the interaction between the different polypeptides must also be well defined to describe the final structure. That is when we get what is known as a quaternary structure. Please note, quaternary structure can be seen only in oligomeric proteins. The quaternary structure can only be seen in oligomeric proteins. Means if I talk about myoglobin, myoglobin has a single polypeptide, and what is the quaternary structure? You will say there is no quaternary structure. Until unless you have more than one polypeptide, you cannot have a quaternary structure. For a quaternary structure, we require more than one polypeptide. For if you have a single polypeptide, then the final structure will be the tertiary structure. For example, myoglobin has a final tertiary structure but not a quaternary structure. Now, another important thing that you should know about when we are talking about orders of protein structure, at this point you should know the different bonds which stabilize V levels. So we'll quickly list the different bonds which stabilize the different levels of the protein organization in the form of a table. Let's quickly draw a table and see Okay, so here we will write the order of protein organization. Here we'll write the order of protein organization. After that, we will write the bonds, bonds which are seen at the different orders of the protein structure. So starting with the primary structure, the bonds that you see in the primary structure are basically the peptide bonds. The only bond that we see in the primary structure is the peptide bond. The only bond that we see in the primary structure is the peptide bond. Remember, the peptide bond is a type of a covalent bond. It is a type of the covalent bond. Moving on to the secondary structure. In the secondary structure, we find the non-covalent bonds. We find the non-covalent bonds. Which are these non-covalent bonds? We can have the hydrogen bond. We can have the Van der Waals forces. We can have the ionic interaction. And lastly, we can have the hydrophobic interactions. Please note the hydrophobic interactions are very important in stabilizing the protein structures. They're very important in stabilizing the protein structures. So in secondary structures, these structures are held together by the non-covalent bonds. Talking about tertiary structure, we have the non-covalent bonds plus the disulfide bonds. Remember, the disulfide bonds are covalent bonds. The disulfide bonds are covalent bonds. And finally, we come to the quaternary structure. In the quaternary structure, we'll have the non-covalent bonds plus the disulfide bonds. In addition, we also have the what is known as condensation bonds. What are condensation bonds? They are cross-linking covalent bonds between the different polypeptides. They are the cross-linking covalent bonds between the different polypeptides. So these are the different bonds that will come across when we are talking about the different levels of protein structure. Now here I'll also mention some secondary structures which generally we get confused about. I mentioned that some of the secondary structures are alpha helix, the beta sheets, the beta pleated sheets, but the loops, turns, and bends are also secondary structures. I'll mention here that the loops, turns, and bends, if you see these terms, they are basically the secondary structures. The loops, turns, and bends are also secondary structures. So just try to understand what is happening. when the protein is translated, it is having the primary structure, the peptide bonds. As the protein structure is being formed, the protein has been translated, the amino acids which are close together will try folding into shapes, giving rise to the secondary structures. Gradually, as the secondary structures start forming, they'll start interacting among themselves and they'll form the tertiary structures. Once the various polypeptides are formed for oligomeric protein, they will finally form the quaternary structure. So basically, this is the progression of the protein to acquire the final shape. Now, in the process of acquiring the final shape, there are two more phenomena which are occurring. In the process of acquiring the final shape, there are two more phenomena which are occurring. One of them is what is known as folding. So the protein folding is also occurring simultaneously. And the second thing which is occurring simultaneously is the post translational modification. Please note that the final protein that you see is not having the exact same composition as what you get after translation, because after translation a lot of modification occurs in the protein. Some amino acids are modified, there is some cross linking, some hydroxyl groups are added, some oxidation can occur. So because of that, the structure or the composition of the amino acids in the protein after translation is modified. The process is known as the post translational modification. The process is known as the post translational modification. So these two are also involved in achieving the final shape. When you're talking about the protein, to achieve the final shape, these two are occurring simultaneously, the folding and the post translational modification. So when we talk about folding, we should remember that the protein itself doesn't know how exactly to fold. It has some idea based on the amino acids, but it cannot try all the permutation and combination, because the number of permutation and combination is very high, so it requires some help. So it gets help from what are known as folding proteins, also known as the auxiliary proteins. The auxiliary proteins assist in protein folding. The auxiliary proteins assist in protein folding. Now, these auxiliary proteins can be of two types. They can be uh, enzymes. So they change, they can cause some isomerization, or they can add some bonds, etc. So they can be some enzymes, or they can be some helper proteins. So two types of auxiliary proteins can be there: the enzymatic auxiliary proteins and the helper auxiliary proteins can be there. Let's take a quick look. Very good, very good, chaperones. Let's take a quick look at some of the important auxiliary proteins that you should know about. Some of the auxiliary proteins that you should know about. The proteins which assist in folding, that is the auxiliary proteins. The first important group are the chaperones. As you rightly said, the chaperones, which can be of two types: the chaperonins and the heat shock proteins. The chaperonins and the heat shock proteins. The protein disulfide isomerase and the peptidyl cis trans isomerase. These are examples of enzymes that I mentioned. These are examples of enzymes that I mentioned. Then we have the calnexin and the calreticulin, very focused for the cartilage and the bone, very commonly seen there. In addition, we have the GRP94, particularly for the transmembrane proteins. We find the involvement of the GRP94 and the immunoglobulin heavy chain binding protein. This is for the folding of the immunoglobulins. This is for the folding of the immunoglobulins. So these are some of the important auxiliary proteins which assist in folding. Some of the auxiliary proteins which assist in folding. To understand the post-translational modification, to understand the post-translational modification, we'll take the example of collagen. Okay, so we've seen the auxiliary proteins how they're assisting in folding, but to understand the post-translational modification, we'll take the example of collagen because collagen shows a very wide variety of post-translational modification. So before I tell you the details of the post-translational modification of collagen, let's briefly take a look at collagen so that we know what molecule are we talking about, and then we'll quickly look at the post-translational modification. So collagen in brief, and then we move on to the post-translational modification. Let's talk about collagen. Okay. First thing to understand, there are various different types of collagen. There are various different type of collagen. So the collagen can be of 28 different types. 28 different types of collagen composed by 32 different genes. So this is the variety that we get to see when we talk about collagen. 28 different types of collagen, and they are synthesized with the help of 32 different genes. Until and unless mentioned or specified, when we say collagen, we tend to mean the type 1. Until unless specified, the collagen means collagen type 1. The collagen type 1 is made up of a triple helix, meaning it has three polypeptides. All collagen will have three polypeptides, but in this case, each of the peptides arranged as a helix. So we have the triple helix in the uh, collagen type 1, and each helix is having a very specific formula, and the formula is glycine XY, repeating n number of times where the n may be as high as 1000. So glycine XY repeating about 1000 times is the structure of each of the polypeptide. So what happens, each of the polypeptide is present as a helix. Okay, so this is the structure that we get in case of the uh, secondary structure that we are looking at. The helix is arranged like this, three helix you can see. So the formula, the structure. In addition, these helices are interlinked. These helices are interlinked with the help of different types of bonds. They can be interlinked with the help of a large number of hydrogen bonds. They can be interlinked with the help of the large number of hydrogen bonds. All right, large number of hydrogen bonds can be there. In addition to the hydrogen bonds, we also find what are known as the covalent bonds. The very stable covalent bonds can also be there. So greatly cross-linked polypeptides are there. The greatly cross-linked polypeptides are there. They are commonly found in the extracellular matrix in a very specific pattern. They are found in the extracellular matrix in a very specific pattern, and we call this pattern as the quarter staggered arrangement. We call this pattern as the quarter staggered arrangement. Every layer is a quarter position ahead of the previous layer. Every layer is quarter position ahead of the previous layer. Okay, this is the quarter staggered arrangement of the collagen. All this arrangement helps to fulfill the final function of the collagen in the extracellular matrix, and that is to provide the strength to the extracellular matrix. That is to provide the strength to the extracellular matrix. See, the quarter stagger arrangement is useful because at any given point if there is a break in the topmost layer, you find at the next three layers the collagen is there to buffer that break. At any point where there is break, you find the other three layers are available to support that break, so the extracellular matrix arrangement doesn't become weak. Similarly, the cross linking gives the extra strength to the collagen, so that these are not actually three different strands. They can be considered as one single strand with this thickness. Because they are so tightly linked, they don't behave as three different strands, rather it behaves at one strand with this thickness. So that is the importance of such high cross linking. In addition, one extra point is each of the helix is right-handed. Each of the helix is right-handed, but they are wound around each other in left-handed pattern. This gives the extra strength. If you try to unwind the uh, individual strand, then the three strands will coil around each other very tightly. If you try to unwind one of the strands from each other, then the coiling of the individual strands will start increasing. So it is very difficult to separate the three strands of collagen. So all this increases the strength of the collagen tremendously, and this is a result of the post translational modification. So let us see what are the post translational modification which enable all these features and that we are seeing. One more point you should know here: the X is proline about 100 to 150 times, and Y is hydroxyproline for a similar number of times. So X and Y are present as proline. One of them is simple proline, the other one is the hydroxylated proline. So let's look at what is happening in the post translational modification of the collagen. So in the cytoplasm, this is where the translation will start. So initiation of translation, and after some time we will see that a structure is formed, a small structure is formed. We call it the signal peptide. We call it the signal peptide or the leader sequence. Now, the leader sequence or the signal peptide is present in those proteins which have to be secreted outside. Please understand that the collagen is present in the extracellular matrix, so it must have been secreted outside. So, we are looking at a protein which has to be secreted, and whether or not it will be secreted depends on presence or absence of signal peptide or the leader sequence. Because as soon as the signal peptide is discovered, translation stops. What happens when the translation is stopped? The migration of translational machinery to endoplasmic reticulum, and the further translation will occur in the endoplasmic reticulum. The further translation will occur in the endoplasmic reticulum, where the translation is completed. It's something like this. You have this endoplasmic reticulum, the sorry, the messenger RNA, the ribosome will come and attach here, forming the rough endoplasmic reticulum, and then the further translation of the protein will occur in the lumen of the endoplasmic reticulum. Further translation of the protein occurs in the lumen of the endoplasmic reticulum. So the translation is completed inside the endoplasmic reticulum, but at the same time, various other changes are also occurring, which we club as the post-translational modification. Number one, leader sequence is no longer required. The leader sequence is cleaved. We don't need the leader sequence, so the leader sequence is cleaved. At the same time, glycosylation occurs, meaning the collagen is a glycoprotein. It is not a simple protein, it is a glycoprotein. There is also hydroxylation of two residues, lysine and proline, by lysyl and prolyl hydroxylase. For this purpose, vitamin C is required as the coenzyme for hydroxylation of lysine and proline. By the hydroxylases, vitamin C is required. Vitamin C is required. And lastly, we see the aggregation of three polypeptides by formation of disulfide bonds. By formation of the disulfide bonds, we get to see the aggregation of the three polypeptides. So once you cleave the leader sequence, the collagen that you are left with is the pro-collagen. The collagen that you are left with is the pro-collagen. The pro-collagen has cysteine rich ends, which I have drawn in red. It has the cysteine rich ends, which I have drawn in red. These are the pro-collagen polypeptides. And as I said, the ends are cysteine rich, though, so they will form the disulfide bonds. So they will form the disulfide bonds. Cysteine rich ends will form the disulfide bonds. At this stage, when the three polypeptides have come together, they will be secreted into the extracellular matrix. They are secreted into the extracellular matrix. Further post-translational modification occurs in the extracellular matrix. So what is happening in the extracellular matrix? The coiling of three polypeptides. This coiling is strengthened by oxidation of lysine by the enzyme lysyl oxidase. For this purpose, now the copper is required. The lysyl oxidase, oxidized lysyl oxidase is going to cross link, or the what we had called the form the condensation bonds between the different polypeptides. Once all this shape is achieved, there is cleavage of the cysteine rich ends. They are not a part of the final structure, so there is cleavage of the cysteine rich ends, and end result what you get is the final functional type 1 collagen known as the tropocollagen. The three oligonucleotides joined together in the extracellular matrix after the post-null modification is the tropocollagen, or you can say it is the collagen type 1. So as you can see, the different covalent modifications that we specified. What are those different modifications? The glycosylation to interact with the cells. The hydroxylation will help to form the hydrogen bonds. It will help to form the hydrogen bonds. Then the oxidation will help to form the cross linking. It will help to form the cross linking between polypeptides as well as the different collagen layers. So all these function help to further increase the strength of the collagen. We had mentioned the different bonds, the hydrogen bonds, the cross linking. All of this is due to the post translational modification of the collagen. So these are some important points about the post-translational modification that you should know about. Because we are talking about the collagen itself, I'll tell you a few important things about collagen here only. So I already said there are 28 different types of collagen which are synthesized by 32 different genes, and we said the most important most common collagen is the collagen type 1. So type 1 is a fibrillar protein. Type 1 is a fibrillar protein, so is a type 2, so is type 3, and so is type 5. Please note that type 4 is a globular protein. Fibrillar, fibrillar, fibrillar. These help us to describe the function of the different collagens. I'll repeat again that the type 1 collagen is the most common collagen in all the tissues. About type 3 collagen, one point that you should know, the type 3 collagen is somewhat elastic, and therefore it is found in those tissues which need to expand. I'll show you where they are found. The globular one is present in the basement membranes. Let's see if we have a slide showing this. Yeah, type 1 present in the most type 1 is present in the most of the uh, connective tissue. Type 2 in cartilage and vitreous humor. The type 3, because it is extensible, it is present in the skin, the lung, the vascular system, meaning the large blood vessels. And type 4, it is present in the basement membrane. It is present in the basement membrane of the kidney, of the internal ear. So it is present in large number of the basement membranes. Okay. Now, there are some important inherited disorders of collagen, the names of which you should know. So, I'll quickly mention some of the important inherited collagen disorders. Some of the important inherited collagen disorders. Please remember, please remember that scurvy is not an inherited Collagen disorder, it is a dietary deficiency disorder. It is a dietary deficiency disorder. Okay. So the first name in this list which you should remember is the osteogenesis imperfecta. Osteogenesis imperfecta. In osteogenesis imperfecta, different types of collagen can be defective because there is six different types, five different types of osteogenesis imperfecta. So type of collagen which is defective will generally not be asked. What you should remember is the blue sclera. What you should remember is the blue sclera. And in these patients the bones are very fragile, so even in childhood, on mild exertion the bones will break, and it gives the presentation of what is known as battered baby. So we call it the pseudo battered baby syndrome. The child will present to the OPD with multiple fractures in different states of healing when it is referred to the tertiary hospital for examination. At the primary and secondary levels, whenever there is a fracture, they will put up a splint or a plaster to help the healing. So after repeated fractures without any injury, the parents will bring the child to tertiary structure, and you find multiple fractures in different stages of healing are there, and you may make a presumptive diagnosis. Okay, this child is beaten up a lot at home, it is a battered baby. But on detailed examination and history taking, you will find no, when the child started crawling, he fractured his radius. When he started walking, he fractured his tibia. Because the bones are fragile, they are not able to take the weight of the body. So the fractures are not due to trauma, they are due to the exertion of walking, and the child is fragile. So osteogenesis imperfecta, one. Second, Ehlers-Danlos syndrome. In Ehlers-Danlos syndrome, also known as the rubber man syndrome, the classical features is hyper flexibility. Very good. Hyper flexibility of joints. Hyper flexibility of joints. The joints are so flexible that they can touch the back of their hand to the forearm. So hyper flexibility of the joints is there. And hyper extensibility of skin. Hyper extensibility of skin. We cannot pull the skin very far, but those individuals are able to pull the skin right up till here. So this is the hyper extensibility of skin. These are two very important features of the Ehlers-Danlos syndrome. Again, the Ehlers-Danlos syndrome is of multiple types. Some of them are risky because this weakness if of the collagen is there in the blood vessels also, and if those patients play contact sports, the large vessels may rupture during the trauma of the sport. So again, Ehlers-Danlos syndrome, multiple types of collagen are defective, so type of collagen will not be asked. Then one of you has written the Alport syndrome. Now, Alport syndrome has been asked a large number of times in the past. In the recent past, it has not been asked, but previously in the AIMS and NEET exam, we have seen the Alport syndrome being asked a very large number of times. So, I'll tell you what you have to know about. Defective collagen is collagen type 4. We already mentioned collagen type 4, which is present in the basement membrane. That is going to be defective. The gene which is defective is called 4A5. The name of the gene which is defective is the call 4A5, which codes for the fifth fibril of collagen type 4. It codes for the fifth fibril of collagen type 4. Each of these is a question. Each of these has been asked as a question. All right, the patient will present with nephritic syndrome. The patient will present with a nephritic syndrome, may be associated with some amount of hearing loss, because the basement membrane in the internal ear may also be defective, so may be associated with some amount of the hearing loss. So collagen 4 gene 4a5 fibril is the fifth fibril collagen 4, and the presentation in the patient is that of the nephritic syndrome. What else? Anything else? We have the disorder called Menkes disease. In Menkes disease, the classical presentation is that of kinky hair, sharply turned or folded hair is there, and there can be flaking of the skin, unhealthy skin can be there, but the classical finding is the kinky hair. In Menkes disease, there is defect in copper transporter. See here, the defect is not in the collagen metabolism, but copper transporter defect is there, resulting in decreased activity of lysyl oxidase. If you recall, lysyl oxidase requires copper. Lysyl oxidase requires copper, and defect in copper transport results in decreased activity of lysyl oxidase. So both the question has been asked. In Menkes disease, what is the primary defect? The primary defect is the defect in the copper transporter. Second question, in Menkes disease, which enzyme is affected? The enzyme which is affected is the lysyl oxidase because of the copper deficiency. The defective enzyme is the ATP7B. Defective transporter is the ATP7B. And then we have the epidermolysis. Bullosa dystrophic type. Epidermolysis bullosa dystrophic type. There are two types of epidermolysis bullosa, simplex and the dystrophic. Here, the defective type that we are dealing with is the dystrophic type, and the defective collagen is collagen 7. Defective collagen is type 7. Okay, some of you are saying 7a. Please check out this 7a 7b in Menkes. uh, as far as I remember, it is 7b, but we have second disorder, Wilson's. So Wilson's and Menkes both have copper transport defect. One of them is 7A, one of them is 7B. I'll check and get back to you, but I think in Menkes it is 7B. Nagendra has, Abhinav Reddy has said it is 7A. I'll just check it out and let you know whether Wilson's is 7A or the Menkes is 7A. Okay, so epidermolysis bullosa dystrophic type, the collagen type 7 is defective. Uh, which is required for the stability of the different layers of the skin. Okay, so these are some of the inherited disorders that you have to remember: the osteogenesis imperfecta, the Ehlers-Danlos syndrome, the Alport syndrome, the Menkes disease, and the epidermolysis bullosa dystrophic type. Some of the important inherited disorders. There are other disorders, obviously, but these are some of the important inherited disorders of the collagen. Some of the important inherited disorders of the collagen. Okay, so if you're saying 7A must be 7A, then 7B should be for Wilson's. Anyway, I'll cross check and verify whether it is 7A or 7B. I'm sorry for the confusion. I just I'm not very sure about the Wilson's and the Menkes. Just came to my mind that the ATP 7B is the transporter. So I'll cross check. Don't worry. Okay, so this is what is being asked from collagen, the inherited disorders, the types, the arrangement, the post-transit modification. One small part which is asked from collagen is collagen in wound healing. The collagen in wound healing, and you should know the what collagen on day 1, what collagen you find on day 3, and what do you find on day 5 oblique day 7. So on day 1 we have the elastic collagen because it has to bridge the gap. On day 1 we have the elastic collagen which has to bridge the gap. So at this point the collagen which is being deposited in the wound is collagen type 3. On day 3 type 1 starts appearing. By day 5 to day 7 all type 3 has been replaced by type 1. So what you find is only the type 1 collagen, only the type 1 collagen uh, on day 5. So in the wound after healing you won't find the collagen type 3. The collagen type 3 will not be there in the wound after healing. It will only be the collagen type 1. This is responsible for scars not being flexible because they don't have any collagen type 3, so they are not elastic. The normal skin is elastic, but if there is a scar it will not be elastic because it doesn't have the collagen type 3. So this again tells you that during healing the previous structure is not exactly restored. The wound healing is not having the same architecture as the original skin. So this is a extra bonus question which is being asked nowadays. You should remember this: the collagen in wound healing on different days, very frequently asked. Okay, so collagen is one of the very important proteins that you should know about. The second very important protein or protein subunit that you should know about is the heme. Okay, can you give me some examples of proteins containing heme? Can you give me some examples of proteins containing heme? At least four examples. Or if you want, you can give more also. Can you give me some examples of heme proteins? Examples of heme proteins. Give me examples of heme proteins. Examples of heme proteins. Please give me examples of heme proteins. Give me some examples of heme proteins. Come on, come on. No examples? Okay, don't worry. I'll give you the examples of heme proteins. Hemoglobin contains heme. Myoglobin, then catalase, then cytochrome, cytochrome C. Cytochrome P450 and tryptophan pyrrolase. Okay, so these are some of the important common heme proteins that you should know about. So this example is just to tell you that when I say heme, it does not just mean hemoglobin. It can be present in large number of other proteins also. This is just to tell you that heme can be present in large number of different proteins also. So let's take a look at the heme. When we talk about heme, we have to talk about two aspects. One is the heme synthesis and second is the heme degradation. Both the parts are important. Reason being, the defect in heme synthesis is associated with porphyrias. The defect in heme synthesis is associated with porphyrias, and the defect in degradation and excretion is related to the hyperbilirubinemias. All right, so we have to talk about the heme synthesis as well as the degradation. All right, please note I have checked and you are correct. In Menkes, the defective transporter is 7A. 7B is for Wilson's. You are right. The 7A is for Menkes. The 7B is the defect seen in Wilson's. Thank you very much for correcting me. So please make this change in your notes. The ATP 7A defective in Menkes for copper transport. In Wilson's will have the 7B. Okay, so let's talk about the heme synthesis and the degradation. Let's talk about the heme synthesis and the degradation. Quickly, we'll take a look at the heme synthesis and then I'll give you the checklist for heme synthesis. Let us quickly go through the heme synthesis and then we'll do the checklist for heme synthesis. The starting material for heme synthesis, succinyl coenzyme A and the amino acid glycine. PLP is the coenzyme for heme synthesis. PLP is the coenzyme for heme synthesis. And for the heme synthesis in liver, for heme synthesis in liver, the rate limiting enzyme is the ALA synthase, only in the liver, not in the bone marrow. And the first step where we are synthesizing ALA, succinyl coenzyme plus glycine giving ALA, occurs in the mitochondria. It occurs in the mitochondria. So what happens in the mitochondria? The succinyl coenzyme A plus glycine will combine to give us delta amino levulinic acid, also known as ALA. The name of the enzyme is the ALA synthase. Please note this is occurring inside the mitochondria. It is occurring inside the mitochondria. Once the synthesis is completed, the ALA will quickly come out. It will quickly come out in the cytoplasm. It will quickly come out in the cytoplasm. Okay, so step one, succinyl coenzyme plus glycine will combine in the presence of ALA synthase to give you the delta amino levulinic acid, that is the ALA. The ALA will come out outside. The two molecules of ALA will combine together to form the porphobilinogen. Two molecules of ALA will combine to form porphobilinogen. The name of the enzyme catalyzing the reaction is the ALA dehydratase. Now the ALA dehydratase is a very important enzyme. 2 points to note here. Aldehyde contains zinc. First point to note, it is a zinc containing enzyme. Second point to note, it is inhibited by lead. So 2 points to note about aldehyde dehydratase. It is a zinc containing enzyme and it is inhibited by lead. So 2 combine to give us the porphobilinogen. 2 combine to give porphobilinogen. The 4 porphobilinogen combine together to form what is known as the hydroxymethylbilane. The 4 PBG combine together to form the hydroxymethylbilane. Okay, the enzyme catalyzing the reaction has multiple names. The enzyme catalyzing the reaction is known commonly as uroporphyrinogen 1 synthase. The other names for the enzyme are porphobilinogen deaminase. Why? Because it is causing the deamination of the porphobilinogen, or the hydroxymethylbilane synthase, because the immediate product which is formed is the hydroxymethylbilane. Any of the three names are used for the same enzyme. Subsequently, the hydroxymethylbilane is converted into uroporphyrinogen 3 by the action of uroporphyrinogen 3 synthase. I'll show all of this in the diagram, don't worry. So next reaction is the conversion of HMB to uroporphyrinogen 3. All right. If the enzyme synthase is not there, HMB will spontaneously cyclize to form the uroporphyrinogen 1. Spontaneously, it will cyclize to form uroporphyrinogen 1. I'll show all this in the diagram where you can make it. So, the 4 molecule of PBG, they undergo the deamination by the uroporphyrinogen 1 synthase, also known as the PBG deaminase. Because HMB is synthesized, this enzyme is also known as HMB synthase. Now, normally the enzyme is there and HMB is very quickly converted into the type 3 uroporphyrinogen. Normally, the enzyme is there and it is quickly converted to the type 3 uroporphyrinogen. However, if the enzyme is not there, if there is a defect, the enzyme is absent, in that case we find the spontaneous cyclization to give us the type 1 uroporphyrinogen. In the previous studies, when we could not freeze the reaction, the end product would be the uroporphyrinogen 1, not HMB, because HMB spontaneously cyclizes. So the old name is the uroporphyrinogen 1 synthase. The end product that you get is the uroporphyrinogen 1, not the HMB. That's why the old name is uroporphyrinogen 1 synthase. However, normally it gets converted to the type 3 uroporphyrinogen. Right. The uroporphyrinogen 3 will subsequently be further metabolized. The uroporphyrinogen 3 is converted into the coproporphyrinogen 3. It is converted into the coproporphyrinogen 3. So what has happened? Reaction 1 occurred in the mitochondria. Reaction 2, 3, 4 and 5 occurred in the cytoplasm. And as soon as the copropeptide 3 is formed, it will enter the mitochondria. It now enters the mitochondria, so that reaction 6, 7 and 8 are again occurring in the mitochondria. Reaction 6, 7 and 8 are again occurring in the mitochondria. Alright, so the copropeptide 3 will now enter the mitochondria. It undergoes two oxidation. First, it is converted to the protoporphyrinogen 3 and then to the protoporphyrin 3. I am showing you these reactions because each of the reaction is associated with the porphyrias. So we must know the reactions, although you need not remember them, but an idea about them is important. The name of the corresponding enzyme for each of them depends on the substrate. The coproporphyrinogen is acted upon by coproporphyrinogen oxidase when you get the protoporphyrinogen 3. The protoporphyrinogen is then again acted upon by the oxidase, the protoporphyrinogen oxidase. Okay, so what we are left with is the protoporphyrin 3, which is then finally converted into heme by the addition of iron. By the addition of iron, the protoporphyrin 3 is converted to heme. The name of the enzyme is ferrochelatase, also known as heme synthase. Also known as heme synthase. So what we get is heme. What we get is heme. Okay, so if you can remember these reactions, good. If you can't remember, doesn't matter. We are going to do the checklist and see what all you should know. Checklist for heme synthesis. First point to note, the heme synthesis occurs in all the tissues because I had shown you so many enzymes and proteins, they are present all over the body. So heme synthesis occurs in all tissues. But here I would add a qualifier, maximum in liver and the bone marrow. So these two tissues will show the maximum heme synthesis, but it does occur in all the tissues. For the compartment, both mitochondria and cytoplasm are involved. Both mitochondria and cytoplasm are involved. The substrates, as we had already mentioned, are succinyl coenzyme A and glycine. Iron in last step. Iron in last step. Okay. End product is obviously heme. End product is obviously heme. The rate limiting enzyme and target for regulation, these are described only for liver, not for muscle. Only for liver, not for muscle, and that is the ala synthase. Only for liver and not for muscle, that is the ala synthase. Okay, so these are the 6 points that you need to mainly remember. In addition, I had given some extra points. They were related to zinc containing enzyme, lead inhibits. Here you should remember lead inhibits 2 ala dehydratase as well as the ferrochelatase, also known as the heme synthesis. This we had written when we are doing the enzymes. Lead inhibits the ala dehydratase and heme synthase. Last thing to remember is 2 ala will give you 1 PBG and 4 PBG will give you 1 hydroxymethylbutylene. This question is also asked, how many ALA join together to form the 1 phosphorylation, how many PPG join together to form the hydroxymethylbutylene. So these are some additional points that you should know. This is a checklist for heme synthesis. Now if any of the enzymes of heme synthesis are deficient, then what we get is the porphyrias. What we get is the porphyrias. Obviously the porphyrias will be discussed in a lot of detail later on, but what you have to know about the porphyrias from the biochemical point of view. The porphyrias have the classical presentation of the triad: abdominal pain, photosensitivity, neuropsychiatric complaint. The acute intermittent porphyria. Acute intermittent porphyria. The AIP does not have photosensitivity because there is no ring structure. There is no ring structure. Okay, there is no ring structure, so there is no photosensitivity. The congenital erythropoietic porphyria. The congenital erythropoietic porphyria and porphyria cutanea, sorry, and protoporphyria, not porphyria cutanea, the protoporphyria. uh Stop. Congenital erythropoietic porphyria and the protoporphyria. These will not present with the abdominal pain. Sometimes the porphyria cutanea tarda may or may not have the abdominal pain. May or may not have the abdominal pain. So that is related to the clinical presentation that you should know from biochemical point. The classical test is the Ehrlich aldehyde test. It identifies porphobilinogen. It is a qualitative test. If porphobilinogen is accumulated, the early childhood test will be positive. Porphobilinogen will accumulate in all the porphyrias. It will accumulate in all the porphyrias. So in the past it was used. Nowadays we don't use this test. Nowadays we use the HPLC. It also tells us the type of porphyria. The early childhood test will not tell us the type of porphyria. It will only tell us that porphyria is there, plus minus, not the type of porphyria. But HPLC will tell us the type of porphyria. And another very important finding is the Soret band. I'll show you the Soret band shortly. So there another additional finding is the Soret band, which is seen in the patients of porphyria. So what is the Soret band? What is the Soret band? When you do the spectral scan of the patients, when you measure the absorption at different wavelengths, in the patients for any body fluid, then you find a graph like this. This is what we find in the normal individual. This is for any body fluid. This can be serum, it can be plasma, it can be urine, it can be synovial fluid, it can be CSF, any body fluid. When you do this measurement, you measure the absorption at different wavelengths, you find a graph like this. But if you measure the absorption in a patient porphyria during an episode, if you try to measure, you get a graph like this. Let's say we're starting at 200 nanometer and going right up till 800 nanometer. So look at a graph like this. This you will see in patient of porphyria during an episode. This peak appears at 400 to 410 nanometer. This peak appears at 400 to 410 nanometer, and this is what we call the Soret band. This is again characteristic of porphyria. This is characteristic of porphyria. The Soret band is characteristic of the porphyria. Okay, so these are some of the point findings that we have to discuss with the heme synthesis. Okay, let's see if we have this graph table. Okay. In the past, you were also asked about the laboratory test based on urinary findings of the different enzymes. But nowadays, we just focus on the HPLC. We just focus on the HPLC of the serum, and it is able to give us a very good result. HPLC is able to give us the good result. The erythropoietic porphyrias The erythropoietic porphyrias generally do not have the abdominal pain that we had mentioned previously, the congenital erythropoietic porphyria and the protoporphyria. The two most common porphyrias would be the Porphyria cutanea tarda and the acute intermittent porphyria. These two are the most common porphyrias, therefore, you must remember the name of the enzymes in these two conditions only. In these two conditions, you must remember the name of the deficient enzyme, because these are the two most common porphyrias. They are likely to land up in your exam. The others, you can forget the name of the enzymes, not required. Okay, so that was about the heme synthesis. Let's now go on to the heme degradation or the heme breakdown. Let us now go on to the heme breakdown. The heme breakdown is a very straightforward process. Very simple, only two reactions are involved in the heme breakdown. First, heme acted upon by the microsomal heme oxygenase forms the hemin, and then it is finally converted into the biliverdin. Heme to biliverdin. We're talking about the heme breakdown now. From heme synthesis, we are moving on to heme breakdown, where the bilirubin will be formed. The heme is converted into biliverdin. So I'll draw it diagrammatically. Okay, heme, see what happens? The heme plus the protein part, it is split, separated. The protein degradation will occur separately. The protein degradation will occur separately. What we are focusing on is the heme degradation. What we are focusing on is the heme degradation. So like I said, heme, it is first converted, it is first converted into biliverdin. It is first converted into biliverdin by the enzyme heme oxygenase. We mentioned the heme oxygenase, also known as the heme oxidase. So what will happen here? The oxygen is required and NADPH is used and the ferric iron with carbon monoxide is released. Ferric iron with carbon monoxide is released. Please note this is probably the only physiological reaction where carbon monoxide is being generated. Only physiological reaction where the carbon monoxide is being generated. Subsequently, the biliverdin is quickly converted. It is quickly converted into the bilirubin. This is a reversible reaction. The name of the enzyme is biliverdin reductase. The name of the enzyme being the biliverdin reductase. So in two simple steps, the heme is quickly converted to bilirubin. Heme is quickly converted to bilirubin. Please understand that this reaction will occur all over the body because heme was being synthesized all over the body. All the tissues contain the heme, so this degradation is occurring in all the tissues. The bilirubin is formed all over the body. However, this bilirubin which is formed is water insoluble. The bilirubin which has been formed is the insoluble form. It is the insoluble form. Therefore, it must be taken to the liver where it can be converted to the soluble form. So now we are going to take this bilirubin to the liver. The bilirubin is transported. The bilirubin is transported by albumin. It is transported by albumin whereby it will reach the liver. Now here I have to tell you very important point. The albumin has two positions where the bilirubin can bind. For that matter, for all molecules which attach to the albumin, there are two positions where they can bind. One is the high affinity and second is the low affinity site. The high affinity and the low affinity site. The high affinity site is able to bind 20 to 25 milligram percent of bilirubin. This number is very important. The high affinity site is able to bind 20 to 25 milligram percent of bilirubin. Why is this number important? Because once this site is saturated, only then the bilirubin will attach to the low affinity site. Once the high affinity site is saturated, only then the bilirubin will attach to the low affinity site. The bilirubin attached to high affinity site will be displaced only at the liver, but the bilirubin attached to low affinity site can be easily displaced, and this is the threshold limit which comes into play when we talk about kernicterus. Because as long as the bilirubin is attached to albumin, it will not be dislodged in the brain. But when the values exceed 20 25 milligram percent in the newborn, when the blood brain barrier is not yet formed, the low affinity site bilirubin will be displaced and will start staining the neural tissue. That is when the kernicterus will occur. So this value of 20 25 milligram percent attached to the high affinity site is very important. So at the liver, with the help of the facilitative transport. Please note the type of transport is the facilitative transport. The facilitative transport is different from passive diffusion. Why? Because in facilitative transport, the transporter is required. When the transporter comes into picture, the process can be saturated. When it is simple diffusion, the process cannot be saturated. The bilirubin will enter the liver. The bilirubin will enter the liver where it is acted upon by the very important enzyme BIL-UGT. So what happens, the albumin will catch hold of the bilirubin from all the tissues all over the body and bring it to the liver. At the liver, the facilitative transport occurs by which the bilirubin will enter inside the liver, where it is acted upon by bil-UGT. Bil-UGT stands for bilirubin UDP, glucuronosyl transferase. So basically what is happening here, to the bilirubin we are going to add the glucuronic acid. To the bilirubin we are going to add the glucuronic acid, and not once but twice. So what we get is, first, the bilirubin monoglucuronide, and then we get the bilirubin diglucuronide. So two glucuronic acid are added one after the other. First bilirubin monoglucuronide, and then we get the bilirubin diglucuronide under the influence of the enzyme bilirubin UDP, glucuronosyl transferase. At this point, the bilirubin diglucuronide is secreted into the bile canaliculi. It is secreted into the bile canaliculi by the transporter protein MRP2. The transporter protein MRP2. MRP2 stands for multi-drug resistance. Multi-drug resistance like protein. The multi-drug resistance protein which is found in the bacteria, the structure is similar to that, so it is called the multi-drug resistance like protein. It belongs to what is known as the MOET, the multi-specific organic anion transporter. All of these questions have been asked. Please note, all of these questions have been asked. Multi-specific organic anion transporter. Okay, so MRP2 is the multi-drug resistance like protein, and it belongs to the class the MOET. It belongs to the family of ABC transporters. ABC transporter stands for ATP binding cassette transporters. All right, so these are the details related to the MRP2 which have been asked over the various years. The MRP2 stands for multi drug resistance like protein. It belongs to the class MOET. MOET is the stands for multi specific organic anion transporter, and the family is the ABC transporter. ABC transporter is a family of primary active transporter. Means this transport is ATP dependent. It doesn't depend on the concentration gradient. It will try to push all the bilirubin diglucuronide into the bile canaliculi. Okay, so till now our bilirubin diglucuronide has reached the bile canaliculi. It has reached the bile canaliculi. From here, it will first go to the CBD. From CBD, it will go to the second part of duodenum. From second part of duodenum, it will go to the small intestine, and from small intestine, it goes to the large intestine, where it is acted upon by the gut flora. Where it is acted upon by the gut flora. Okay, in the large intestine, it is acted upon by the gut flora. The gut flora will carry out some changes which are listed here. The gut flora will carry out some changes which are listed here. They are: number one, deconjugation. In the liver, the conjugation has occurred by the bil-UGT. Here, some of the gut flora will deconjugate some of the bilirubin. In addition to deconjugation, the conversion to stercobilinogen and urobilinogen. Conversion to stercobilinogen and to urobilinogen. Out of this, a small amount, of urobilinogen is absorbed. How much? About 10 to 15%. 10 to 15% is absorbed and reaches back to the liver. This is known as enterohepatic. This is known as enterohepatic circulation, from liver to the intestine and back to the liver. So this is the enterohepatic circulation. Out of this 10 to 15%, about 5 to 10%, out of this 10 to 15%, about 5 to 10% remains in the blood, and because it is the conjugated form, from here it is excreted in the urine. So in urine we find small amount of urobilinogen normally, which is undetectable. Normally we find a very small amount of urobilinogen which is undetectable. Only when the urobilinogen is high we will be able to detect it. Lastly, the leftover deconjugated bilirubin, the stercobilinogen and the urobilinogen, will be excreted in the stool. It will be excreted in the stool. When the stool gets exposed to air, gets exposed to the air, the stercobilinogen gets converted into the brown pigment which is responsible for the color of the stool. It gets converted to the brown pigment which is responsible for the color of the stool. So I'll quickly recap all of this. No, it has become too small. Okay, so the heme protein will split into two. It will split into the protein which is degraded separately and the heme part. The heme, under the influence of the enzyme heme oxygenase, is first converted to biliverdin, and biliverdin is then converted to bilirubin. This conversion occurs all over the body. This conversion is occurring all over the body. All right. The bilirubin which has been formed is the water insoluble form. Therefore, it has to be conjugated. For conjugation, it is brought to liver by the albumin. Albumin attaches the bilirubin first to the high affinity site. When it is saturated, then it attaches to the low affinity site. At liver, via facilitative transport, the bilirubin enters the liver, where it is acted upon by the enzyme bilirubin UGT, which stands for bilirubin UDP glucuronyl transferase, whereby first the bilirubin monoglucuronide is formed and then the bilirubin diglucuronide is formed. The The bilirubin diglucuronide is actively secreted into the bile canaliculi by the action of the transporter protein MRP2. MRP2 stands for the multi-drug resistance like protein. It belongs to the class MOIT. MOIT stands for multispecific organic anion transporter, and the family is ABC transporter, ATP binding cassette transporter. It is a family of primary active transporters. So the bilirubin diglucuronide will enter the bile canaliculi. From here it reaches the common bile duct. From here it reaches the second part of duodenum, and then the small intestine, and finally the large intestine. In the large intestine, it is acted upon by gut flora, whereby three primary changes occur: deconjugation of bilirubin, the conversion to stercobilinogen, and the conversion to the urobilinogen. These are the three primary changes which occur in the large intestine under the influence of the gut flora. Some amount of urobilinogen, about 10 to 15%, is absorbed and reaches back to the liver. This is known as the enterohepatic circulation. Out of this 10 to 15%, 5 to 10%, 5-10% of 10% stays in the blood, and because it is the conjugated form, water soluble form, it can be excreted in the urine. The deconjugated bilirubin, the stercobilinogen, and urobilinogen are lost in the stool. When the stool is exposed to air, the stercobilinogen gets oxidized to the brown pigment, which is responsible for the color of the stool. Till now, in the breakdown, if anything is unclear, please tell me. Till now, in the breakdown of heme, if anything is unclear, please tell me. Otherwise, I'll go to the other details related to the bilirubin. Okay, let's go on. Sometimes there can be suddenly large amount of bilirubin being formed. This typically occurs when there is hemolysis. When there is hemolysis, suddenly large amount of bilirubin is formed. All of this bilirubin cannot be taken up by the facilitative transport because it will quickly get saturated. So the bilirubin level will rise. The problem here is occurring before the liver. We call it the pre-hepatic jaundice. Sometimes the problem can be at the level of liver, the conjugation is not occurring, or the secretion is not occurring, so it will be the hepatic jaundice. And finally, if after conjugation, if there is a block in the secretion, or if there is a block here, or if there is a block here, in that case we call it the post hepatic, which is mainly due to obstruction, so also known as the obstructive type of jaundice. So the prehepatic, hepatic, posthepatic. Prehepatic is mostly hemolysis related, so it is hemolytic jaundice, and the posthepatic is mostly obstructive jaundice. In each of the condition, we have to comment on two parameters in two domains. What have to talk about? We have to describe the status of bilirubin. We measure the bilirubin in blood and urine, and we measure the urobilinogen. We measure the urobilinogen in urine and stool. The bilirubin that we measure in the blood is total, direct, indirect. Okay, so we are looking at four parameters: bilirubin in blood, the bilirubin in urine, the urobilinogen in urine, and the urobilinogen in stool. So these are the four parameters that we are looking at. So I'll give you a table showing these four parameters in the different conditions in the prehepatic, hepatic, and post-hepatic. Let's take a look. There's something wrong with my chat. Let's see. I'm not giving any questions. It is unlikely. Oh, great. Okay. All this we have done. All this we have done. Okay. So this is the prehepatic, hepatic, and post-hepatic. The prehepatic, hepatic, and the post-hepatic, and this is the table that you are going to draw. The table that you are going to draw. Okay, so like I told you, the parameters are the serum bilirubin, the urine bilirubin, the urine urobilinogen, and the fecal urobilinogen. These are the parameters. These are the parameters that you have to note. Normally, the values are serum bilirubin less than 1, urine bilirubin is absent, not detectable, urine urobilinogen is again not detectable, 0 to 4 milligram, very difficult to detect. The fecal urobilinogen 40 to 80 milligram per 24 hours. When you come to hemolytic anemia, the bilirubin is elevated. It is the indirect type of bilirubin which is elevated. The urinary bilirubin will be absent, the urine urobilinogen is increased, and the fecal urobilinogen is increased. First you note down and then I'll tell you how the changes are occurring. In hepatic jaundice, both direct and indirect bilirubin will be elevated. The urinary bilirubin is present if micro obstruction occurs. The urine urobilinogen is again decreased if microobstruction is present, and fecal urobilinogen is decreased. Coming to obstructive jaundice, the bilirubin which is elevated is the direct type. The urine bilirubin is present. The urine urobilinogen is absent, and fecal urobilinogen is a trace to absent. Let's see what is happening. Let's first talk about the hemolytic or the prehepatic jaundice. Hemolytic or the prehepatic jaundice. Here we had said suddenly large amount of bilirubin is being formed. Large amount of bilirubin is being formed. So this is the water insoluble form. Indirect is the insoluble form. It is the insoluble form. So it will start rising. So the type which is elevated is the insoluble form. Because it is insoluble form, it cannot appear in the urine. Only the soluble form will go in the urine. It is insoluble, so it will not go into the urine. It will not go into the urine. So what is happening now? A lot of bilirubin is being formed. So liver will try to take up as much bilirubin as possible, and this excess bilirubin will be conjugated and it will be secreted in the intestine. Excess bilirubin is there, so excess urobilinogen will be formed, and because excess urobilinogen is formed, so it will be absorbed and secreted in the urine. All right. So this excess urobilinogen will be seen in the stool also. In obstructive, the opposite thing happens. In obstructive, the conjugation has occurred, but after conjugation, the secretion is not occurring. So the form that you see in the blood is the conjugated form, which is the soluble form. The direct is the conjugated form. Indirect is the unconjugated form. This soluble form or the conjugated form can be excreted in the urine because it is water soluble. It can be excreted in the urine. However, because the obstruction is there, the bilirubin is not going into the small intestine. If it doesn't go into the small intestine, it will not reach the large intestine. The urobilinogen cannot be formed, so it is not there in the stool, it is not there in the urine, because if it is not formed, how can it be there? In hepatic jaundice, there is a phenomena called micro obstruction due to edema of the cells. When the hepatocytes will swell due to the inflammation, they will block the canaliculi. If there is canaliculi blockage, we call it micro obstruction. So depending on inflammation, the urinary bilirubin may be present if micro obstruction is there. If micro obstruction is there, the same thing as obstructive jaundice. So same features will see decreased if the micro obstruction is there, and the urinary bilirubin will be there if the micro obstruction is there. Okay, some additional points at this juncture. All of this related to the acquired form of jaundice. All of this relates to the acquired form of jaundice, but we also have what is known as the inherited types of jaundice. We also have inherited hyperbilirubinemia. We also have what is known as the inherited hyperbilirubinemia. Okay, in the inherited hyperbilirubinemia, we have two types: the unconjugated hyperbilirubinemia and the conjugated type of the hyperbilirubinemia. In the unconjugated, the names that we have to remember are Krigler-Najar type 1, Krigler-Najar type 2, and the Gilbert syndrome. And the Gilbert syndrome, right? In all of these conditions, the defect, very good, the Krigler-Najar syndrome and the Gilbert syndrome, the defect in all of these conditions is in the conjugating enzyme. So what is the defective component? The bil UGT is the defective component. The bil UGT is the defective component. If you look at the activity in Krigler-Najar, the activity is 0%. In type 2, the activity is less than 5%, and in Gilbert syndrome, the activity is less than 30%. So basically, they are the spectrum of the same disease. A point to remember: the bil UGT can be activated by the phenobarbital. Alright, so in these two conditions, if you give the phenobarbital, there will be the symptomatic improvement because bil UGT activity will increase, and therefore it will try to clear out some of the bilirubin. It will try to clear out some of the bilirubin. Okay, so this is the unconjugated hyperbilirubinemia. In conjugated, can you give me the names? In conjugated, can you give me the names? The rotors and the Dubin Johnson. The rotors and the Dubin Johnson, these are the conjugated types, the rotor and the Dubin Johnson. In both of these, the defect is in the secretory protein. Defect is in the secretory protein, the MRP2. Please remember, among all these conditions, most severe jaundice is seen in which condition? Most severe jaundice is seen in which condition? The answer is the Krigler-Najar type 1. The most severe jaundice is seen in the Krigler-Najar type 1. Okay, the activity of MRP2 is also increased by phenobarbital, so we can give the phenobarbital also in rotors and Dubin Johnson. So conjugation as well as secretory activity both increased by the action of the phenobarbital. Now if you carry out the electrophoresis, of the bilirubin, if you carry out the electrophoresis of bilirubin, you are going to get four fragments. We are going to get four fragments which we label as alpha, beta, gamma, and the delta fragments. We're going to get the four fragments labeled as alpha, beta, gamma, and the delta fragments. The alpha fragment is the unconjugated bilirubin. The alpha fragment is the unconjugated bilirubin. The beta fragment is the bilirubin monoglucuronide. The gamma fragment is the bilirubin diglucuronide, and delta fragment is the bilirubin albumin conjugate. Now you'll ask, what is this bilirubin albumin conjugate? Bilirubin albumin conjugate is covalently linked bilirubin. Normally the linkage is non-covalent, but covalently linked bilirubin and albumin seen in chronic jaundice. The covalently linked bilirubin and albumin seen in cases of chronic jaundice. All right. So these are the different fragments. The difference between the different fragments is after treatment, the delta fragment disappears very late. Reason being, now it is attached to the albumin and it has the same half-life as that of the albumin. What is the T half of albumin? It is 19 to 21 days. So the half-life of this delta fragment is also 19 to 21 days. Others will have the half-life of 24 to 36 hours. The others will be quickly excreted, but the delta fragment has a very large half-life of 19 to 21 days because now it is attached to the albumin. The last thing that you should know, is under the heading of the unconjugated inherited hyperbilirubinemia, and that is what is known as the Lucy Driscoll syndrome. Can anyone tell me what is Lucy Driscoll syndrome? The last point that we're going to discuss today is the Lucy Driscoll syndrome. What is Lucy Driscoll syndrome? Anyone can tell me what is the Lucy Driscoll syndrome? Tell me what is the Lucy Driscoll syndrome? The Lucy Driscoll syndrome, anyone, is described as the transient neonatal unconjugated hyperbilirubinemia. It is a transient neonatal unconjugated hyperbilirubinemia. What is happening here? We have the inhibitor of bil UGT in mother's milk. This inhibitor is a mild inhibitor, a mild inhibitor of bil UGT in mother's milk. So it prevents conjugation of bilirubin in first few weeks when system is immature. It will disappear on its own. It will disappear on its own. You don't need to give any treatment. Only thing is you have to watch out for the elevation of bilirubin. If it goes into the danger zone, then you have to give the phototherapy. You have to give the phototherapy whereby the insoluble bilirubin will be converted to the insoluble bilirubin can be converted to lumirubin, which is water soluble. It is converted to the lumirubin, which is water soluble. So only thing you need to do is, if when the level rises beyond 20 milligram percent, then you have to give the phototherapy. The management of hyperbilirubinemia will be discussed in the pediatrics in the neonatal jaundice. It is one possible cause of breast milk jaundice. It is not the only cause of breast milk jaundice, but it is one of the possible causes for the breast milk jaundice. Alright, it is one of the possible causes for the breast milk jaundice. Okay. So phototherapy is the initial approach. If the bilirubin rises even higher, then you may even need to do the exchange transfusion. If bilirubin is greater than 20 to 25 milligram percent, in that case you have to do the exchange transfusion. Details will obviously be discussed in your uh, neonatology section and pediatrics, but just for reference, first phototherapy. If phototherapy is not enough, then you go for exchange transfusion for treatment of the hyperbilirubinemia in the newborn. In adults, the hyperbilirubinemia per se is not a problem because the blood-brain barrier is intact. Kernicterus will not occur. The kernicterus occurs only in the neonates because the blood-brain barrier is still not developed. All right. So all this, the phototherapy and the exchange transfusion is required only in the neonates. In adults, it is not required. Okay, so that completes our discussion on the heme breakdown and the bilirubin. I have tried to cover all the questions that are being asked from the bilirubin section. They are being asked in the pediatrics, in medicine, in surgery, in pathology, in pharma, in physiology. Everywhere the questions are coming, and I've tried to cover all of them. If you have any doubts, if you have any questions, please feel free to ask. Any questions so far? Because if you don't have questions, then I'll start asking questions. No questions? If there are no questions, then I'll start asking questions. If you have any doubts, first clarify your doubts. You started giving the answers. If you have any doubts, first tell me your doubts. If you have any questions, any part you want me to repeat, tell me that first, then we'll go to the questions. Okay. The heme synthesis requires iron, it requires glycine, it requires succinyl coenzyme, it does not require alanine. So alanine is the correct answer. Very good. Succinyl coenzyme and glycine required in the first step, iron required in the last step by the heme synthase. Pigment providing a color to the stool. Your options are the stercobilinogen, the urobilinogen, the mevobilirubin, and the bilirubin. The stercobilinogen, urobilinogen, mevobilirubin, and the bilirubin. The correct answer is the stercobilinogen. It gets oxidized to the brown pigment. Very good. Porphyrogen is formed from how many molecules of ALA? See, I've said this conjugation part will be asked when the condensation is occurring. So you have to give this answer. Porphyrogen is formed from how many molecules of ALA? Your options are 1, 2, 3, and 4. What is the correct answer? Porphyrogen is formed from condensation of two molecules of ALA. Two molecules of ALA combine to form one porphyrogen, and 4-porphyrogen will combine to form one hydroxymethylbutylene. This we had discussed. Zinc containing enzyme are all except. Now sometimes you'll get question where you don't know about all the options, but in that case you should be able to eliminate the options. Remember we talked about the two enzymes ALA dehydratase and ALA synthase, right? Talked about ALA dehydratase and ALA synthase. I said ALA dehydratase contains zinc, and I did not mention about ALA synthase. This means the ALA synthase does not contain zinc. It also means that dehydrogenase and the carbonic anhydrase also contain zinc. Remember, carbonic anhydrase, alcohol dehydrogenase are zinc containing metalloenzymes. They are zinc containing metalloenzymes. In hemolytic anemia, which of the following is not raised? Which of the following is not raised? Your options are serum bilirubin, the urinary bilirubin, the urinary urobilinogen, and the fecal stercobilinogen. Okay, I'll wait for the answer. In hemolytic anemia, which of the following is not raised? This will tell me whether you have understood what we have discussed or not. Whether the understanding is clear or not will become apparent very soon. Okay. What about others? Pooja has given us an answer. What about others? What about others? What do you think should be the answer? B, 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 B. Very good. See, in hemolytic anemia, what we have is unconjugated bilirubin, which is water insoluble, so it will not reach the urine. Urinary bilirubin is the correct answer. Very good. Urinary bilirubin is the correct answer. In all jaundice, the serum bilirubin will be elevated. Whether it is conjugated or unconjugated is a different story, but bilirubin will obviously be elevated in all types of jaundice. Now, I have not talked about this, but you already know this. I am sure you already know this. All of the following enzymes are raised in hepatic jaundice except The ALT means alanine transaminase. AST is the aspartate transaminase. ALP stands for alkaline phosphatase. So what is the answer? All of the following is raised in hepatic jaundice except all of the following is raised in hepatic jaundice except what is the answer? In hepatic jaundice, the alanine transaminase and the aspartate transaminase, which are hepatic enzyme, will obviously be elevated, and because it is jaundice, the serum bilirubin will definitely be elevated. Alkaline phosphatase is elevated in obstructive jaundice. Which which other enzymes are elevated in obstructive jaundice? Which other enzymes are elevated in obstructive jaundice? Please note the names: 5 dash nucleotidase and the GGT, the 5 dash nucleotidase and the GGT. GGT stands for gamma glutamyl transpeptidase. The gamma glutamyl transpeptidase. Very good. GGT and the 5 dash nucleotidase. Conjugation of bilirubin in liver is done by addition of which of the following molecules: acetate, the sulfate, the methyl group, or the glucuronic acid? The correct answer obviously is the glucuronic acid. Glucuronic acid added by the UGT, bilirubin UDP glucuronosyl transferase. Now this is a very important question. I'll wait for you to give me the answer. Most of the bilirubin in bile is alpha bilirubin or beta bilirubin or the gamma bilirubin or the delta bilirubin. If you recall, we had mentioned the alpha bilirubin is the unconjugated bilirubin. Beta is bilirubin monoglucuronide. Gamma is bilirubin diglucuronide and delta is bilirubin albumin conjugate. So let's see what you're going to answer. Murtaza, very good. Anyone else? Okay. The most of the bilirubin in bile is the bilirubin diglucuronide secreted by the MRP2. Recall, MRP2 will secrete the bilirubin diglucuronide, which is the gamma bilirubin. So the correct answer is the gamma bilirubin. Very good. The correct answer is the gamma bilirubin. Supriya, AK, Misala, Srinivas have given the correct answer. Very good. In obstructive jaundice, the color of the stool is green, black, mustard color, also known as deep yellow, or the clay colored or the pale colored stool is there. See what happens in obstructive jaundice? No bilirubin in intestine. Because there is no bilirubin, no stercobilinogen. Because there is no stercobilinogen, there is no brown pigment. So what we get? We get the pale or clay colored stool. We get the pale or clay colored stool. If I change the question from stool to urine, in that case what will be the answer? In urine we will get the mustard color or deep yellow, because all the conjugated bilirubin will be lost in the urine only. All the conjugated bilirubin will appear in the urine, and they will get the mustard color or deep yellow. When we talk about the urine, we'll get the mustard color or the deep yellow. In hemoglobin, the iron is bound to histidine. This I'll tell you, the iron is bound to histidine, and that too the proximal histidine. We have two important histidine in the iron, in the heme, the proximal and the distal histidine. The hemoglobin iron is bound to the proximal histidine. So that is all about the heme metabolism, the collagen and the proteins that we've discussed today. Tomorrow we'll take a quick look at the lipids, and after we complete the lipids, we move on to the molecular biology section, which is the last section of our discussion. So far, if you have any questions, any doubts, please feel free to let me know. Otherwise, we'll stop here today. And again, tomorrow we'll continue with the lipid section. Tomorrow we are going to start with the lipid section. Any questions or doubts so far? Anything you want to ask me? Any part you want me to repeat? Please let me know. If there are no questions, if there are no doubts, I think we will sign off for today. Please send your replies that it's okay. Just give a thumbs up or a good night, so that we know that we can close the session today. Okay. I'm quickly repeating the heme synthesis as requested by Pooja. Let's quickly take a look at the heme synthesis. In the mitochondria, the first reaction is occurring where succinyl coenzyme and glycine will combine to form the ALA. ALA will come out, and it will combine with another ALA. It will combine with another ALA in the presence of ALA dehydratase. It will combine with another ALA in the presence of ALA dehydratase to give you the PBG, porphobilinogen. This PBG will combine with three more PBG. Under the influence of the enzyme Uroporphyrinogen 1 synthase, also known as the hydroxymethylbilane synthase, also known as the PBG deaminase, to give us the hydroxymethylbilane. The hydroxymethylbilane spontaneously cyclizes to form uroporphyrinogen 1. In the presence of the enzyme uroporphyrinogen 3 synthase, yes, एक minute, it forms the uroporphyrinogen 3. Alright, so it will form the uroporphyrinogen 3 if the enzyme is there. If enzyme is not there, it forms the uroporphyrinogen 1. The uroporphyrinogen 3 will get converted into the coproporphyrinogen. Okay, till now the reactions, please note the reaction number 2, the reaction 3, reaction 4 and reaction 5, they occur in the cytoplasm. The coproporphyrinogen will now re-enter. The coproporphyrinogen will now re-enter the mitochondria, where it will undergo oxidation twice. By the enzymes oxidases. If you don't remember the names, doesn't matter. And finally, there is addition of iron by heme synthesis. So the intervening enzymes, even if you don't remember, it is okay. This is the uroporphyrinogen carboxylase. After that, we have the oxidases, and then we have the heme synthesis, which gives you the final heme. Okay. Any specific part, Pooja, that you wanted clarification about? I've quickly done the recap. I've skipped out some of the less important reactions, particularly the oxidases. But any specific part that you wanted me to highlight? We had mentioned the aldehyde is the zinc containing metalloenzyme. The others are carbonic anhydrase and alcohol dehydrogenase. बस 2 मिनट हो गया 2 मिनट Anything else? So we'll stop the transmission.